Hello and welcome to another exciting lecture for Foundations of Behavioral Neuroscience. In this lecture, we will be talking about psychopharmacology. It'll be the first half of Chapter 4. We're going to start with an overview of psychopharmacology. Then we're going to look at pharmacokinetics, drug effectiveness, effects of repeated administration of drugs. We'll look briefly at the placebo effect. Then we'll look at agonists and antagonists when it comes to these types of drug effects. And finally, we'll finish up with a brief bit about addiction. Let's start with a video.
The wood spider is the most accomplished of all web-building species. Recently, scientists gave these tiny creatures a variety of psychoactive drugs to observe their effects on web building. When given a minute dose of LSD, the spider's web took on an unfamiliar, minimalist structure. When given caffeine, the web structure was not affected, but the spider's behavior was. Given THC, the active ingredient in marijuana, the spider didn't build a web. It built a hammock, where it lay all day and watched the caffeine spider go. When given alcohol, the spider built a web, found a mate, and raised over 100 young. But the mate got a restraining order, and now the spider can't go within 100 centimeters of the web. The crack cocaine spider figured building webs was for suckers, waited till the caffeine spider was exhausted, then came up behind it and popped a cap in its ass. Nice web, Mr. Crack Spider. When winter came, the marijuana spider had no place to live. It ended up in the crack web as the crack spider's bitch. For more information on the crack spider's bitch, contact the Canadian Wildlife Service in Ottawa.
Okay, so amusing video. The beginning bits, though, were relevant. The beginning bits were true in how they reacted to it. Obviously, then it devolved from there. But the point is that these different drug effects, these different drugs, have different drug effects. They have different effects on behavior. And that is what we'll get into with this set of slides. So let's start with an overview of psychopharmacology. So psychopharmacology is the study of the effects of drugs on the nervous system and behavior. It is a subcategory of pharmacology, which is the study of all drugs. And when we look at this, it is basically going to be just drugs that affect the central nervous system and the peripheral nervous system as well as behavior. So some drugs that don't necessarily affect behavior will fall under psychopharmacology. There are two major aspects of drug influence, and you notice I've got three here, but let's look at these two major factors or aspects. First, we've got drug effects, which is pharmacodynamics. This is going to look at the effects of drugs. So it's a subcategory under psychopharmacology as well as a subcategory under other drugs, but it is basically looking at the effects. The other is the sites of action. This is where does the process occur that results in the effect. So when we look at the effects, the sites of action are important, and we'll talk about this. We should also bring up another term, pharmacokinetics, which is the process by which drugs are absorbed, transported, metabolized, and eliminated. And we are going to look at that in detail. So let's start with that, pharmacokinetics. Pharmacokinetics is, again, it's this four-step process. We start with absorption here. And absorption is how the drugs are administered. And we'll look at that. We'll look at routes of administration. But it's basically how they're administered and absorbed into the system, whether it's through tissues, into the bloodstream, nasally, if I can talk, all of those, that's relating to absorption. Then the next component is distribution. Distribution is how it's distributed through the body and the blood. And we'll go into this very briefly, but not too much into distribution. But then we'll look at metabolism, which is where the drug is changed to an inactive form by enzymes. This usually happens in the liver, but it can happen in the blood too. And then the final one, the one we're not going to really talk about too much, is excretion, and that's just how it's excreted by urine by the kidneys. Okay, let's start with these. Let's start with the absorption. But to talk about absorption, we have to look at routes of administration. And there are many. I'm only actually putting a few here, but some of the more common ones, like intravenous, where it's put into veins. This is the fastest. It gets pretty much within seconds, it can get to the brain. This is one, though, that takes work. It takes care to be able to do it correctly. There's always inhalation, smoke, stuff like that, where you get like tobacco, nicotine, weed, stuff like that. There's intramuscular. This is where the injection is put into the muscle and then it's absorbed from the capillaries in the muscle into the bloodstream. It's a little bit slower, but it's one where it's easier to do. When you got your COVID vaccine, it was done intramuscular, so it was put into your arm muscle. You could have subcutaneous, which is basically where it's going underneath the skin. Intranasal, which is going where you're breathing it nasally. Or even oral. The problem is, with oral, is the stomach breaks down a lot of things, so some things cannot be done orally. There's other ones I don't have here, like ones where you put it under the tongue and it's absorbed into the bloodstream that way. There's topical, where it can be basically put on the skin and it's absorbed through the skin. The point is there's many ways of administration. Okay, next we'll talk about distribution. Distribution is basically where the drugs enter and spread throughout the nervous system, and specifically mostly on the central nervous system in the brain. And the type of drug it is will actually affect this, because if the drug binds to lipids, to fats, it actually can cross the blood-brain barrier. If it's water soluble, it can't cross that blood-brain barrier as easily, so it is something that won't have as much of an effect. So two drugs that essentially do the exact same thing, if one is lipid soluble and another is water soluble, the lipid soluble one is going to have a greater effect because it's going to get to the sites of action more. The sites of action are where the drugs exert their effect, and it's going to exert its effect on the neurotransmitter system. Later on, we'll talk about agonists and antagonists and all these effects that these different drugs do, but basically all these drugs happen at sites of action where these agonistic or antagonistic effects will occur. The final two are metabolism and excretion. The metabolism one is actually pretty interesting. So many drugs are metabolized and deactivated by enzymes, mainly in the liver, but also in the brain. Then all drugs are eventually excreted, mostly by the kidneys, sometimes by respiration, things like that. But an important thing to consider here is that enzymes actually sometimes transform molecules into more active versions. Some drugs get broken down into other drugs that are actually more active than the original. It can actually cause an extended effect where the enzymes are breaking the drugs down, but because they're breaking them down into something that's also active or even more active, it results in a prolonged effect. Let's transition now into drug effectiveness. So I'm going to talk really long about effectiveness as a whole, but really what we're talking about when we're talking about effectiveness is that we measure a wide variance of effectiveness, or what the drug does to the system. And an important thing to consider when we're talking about this going forward is some drugs, and such as opiates, have more than one effect. They could have a analgesic effect, and they could have a depressive effect on the heart rate, respiration, these types of things. So when we're looking at these and when we're measuring effectiveness, you have to be aware that many drugs actually have multiple effects. And when we're talking about the different things, when we're looking at like how much it takes, and when we come into things like tolerance and all of that, you have to take into consideration that these different effects actually vary in a lot of different things, not just how they affect the body, but how the body responds to them. But we'll come back to that. Let's look at a dose-response curve. So a dose-response curve is basically talking about how much of the drug you need to get a full effect or to get a high effect. Sometimes you only need half of an effect from a drug. But the point is as the dose goes up, the effect goes up. However, the big caveat is there's a ceiling. There becomes a point where increasing the dose doesn't produce a stronger effect. This tends to be due to the fact that the system is flooded with it, that it's already doing the maximum it can. So increasingly stronger doses of a drug produce increasingly larger effects until the maximum effect is reached. And then after that, increments more of the drug do not produce any increase in the drug's effect. However, this could increase side effects because, as I just said on the previous slide, most drugs don't have just one effect. They have multiple effects. So this effect right here could be the effect that we're trying to get out of a drug. However, the ceiling for some side effects, such as the depressive effects on heart rate with opiates, could have a much higher ceiling or no ceiling at all. Or basically the ceiling is so high that it's going to kill you before it reaches the ceiling. So this down here, where it's kind of flat here, this is where not enough of the drug is gotten in the system to have any effect. Then it starts rising due to the increased dose, increased effect, until we get to this flat point up here where we've reached the ceiling. Let's say we're trying to measure effectiveness. Well, one way we measure effectiveness is with what's called the therapeutic index. The therapeutic index doesn't just measure, though, the drug itself, it measures the drug and the drug's side effects. So it is, in a sense, becomes a measure of the drug's margin of safety. It measures how much of the drug needs to be administered to get an effect compared against how much of the drug will result in a deleterious effect. And when we're looking at this, we also have to consider the fact that there's reasons why drugs vary in effectiveness. The first is the sites of action. Some sites of action are going to result in a drug having more of an effect. Some sites are going to have less. And then the activity of a drug with the site of action. And this is what we're going to talk about at the end when we talk about agonistic and antagonistic effects. All right, let's look at the first one though. Let's look at therapeutic index here. To look at therapeutic index, we need two factors. First, we need what is an effective dose. So there is a curve, just like our dose response curve from before. There is a dose response curve that is going to look at the dose at which 50% of subjects show the desired effect. So that is how the dose response curve is going to be mapped for what is an effective dose. 50% of subjects show the desired effect. Then another dose response curve is going to be mapped with the lethal dose. So the lethal dose is going to be the dose at which 50% of subjects die. So that is a curve in which 50% of subjects die. It's actually only examined in preclinical studies, and it's typically only done in animal models. It's definitely not something ethically done in human models. It's more of a theoretical. And it's important to note that doses for both of these are normalized for body weight because body weight affects the spread of the drug in the system. So earlier when we talked about metabolism and distribution, well, if you're bigger, there's more of the body to distribute the drug to, meaning there's going to be less at the sites of action. So when we do this therapeutic index, we draw out, again, the effective dose curve and the lethal dose curve, and essentially look at the margin between them is referred to as the margin of safety. And this is a magnitude thing. So if the lethal is 2 times the dose of the effective, then it is given a rating of 2. And it's a magnitude. It's not given exactly a rating of 2, but this is a magnitude measure. And basically, the larger the magnitude, the better, because then it is more safe to administer a little bit extra. If you've got a drug that doesn't kill you until you've basically taken a pound of it, and you need to take a little pill, well, taking 2 or 3 little pills isn't going to hurt. A good example of this, just off the top of my head, because I've been having to deal with this with dogs lately, is heartworm medicine, heartworm preventative for dogs. So flea and tick and heartworm preventative for dogs. The overdose level is essentially so large that you're never going to get to it. It would be virtually impossible for you to get your dog to eat that much of it. So when you're talking about the effective dose, you measure dose by the weight of the dog. But if you've got a dose for a 40-pound dog and you've got a 20-pound dog, you can give the 40-pound dose to the 20-pound dog because it's not going to adversely affect them because the overdose factor is so high. The margin of safety is so large that you're not going to get to the overdose. So, and the only reason you should get the kind that's for your weight of your dog is because it's cheaper. So the dose for a 20-pound dog is cheaper than the dose for a 40-pound dog. But if all you've got is a dose for a 40-pound dog, then you can give it to a 20-pound dog without worrying about it. So when you've got a higher index, it's a lower level of risk of fatal overdose, but a lower index is a higher risk. So when we look at things like, let's say, barbiturates, which were early depressors, they had a very small margin of safety. We're talking about where it was really easy to overdose. It's why barbiturates aren't really prescribed anymore, because there's other drugs now that do the exact same thing that's margin of safety is magnitudes higher. You're looking at margins of safety in the range of 100 times. So you've got to get 100 times the effective dose to get to the lethal dose, versus barbiturates that tend to have about 2 to 3 times the safe dose before you overdose. Very close, very small margin of safety. So let's look at the dose response curve for morphine. So the dose response curve for morphine, again, you've got the effective here, the lethal here, and then you've got this margin of safety. What it actually is the analgesic effects of morphine are at this point. So that's where the effective dose is, because that's what morphine, it's to dull pain, various different things like that. But the point is this is where it's effective at. However, the dose response curve, which is different, you're going to have a dose response curve that's different for each of the different effects. That's important to note. Each effect has a dose response curve. Well, the dose response curve for the depressive effects of morphine on respiration is over here. And why is that the margin of safety? And because once you've depressed the respiratory system so much, it ends in death, results in death. So that's the margin of safety, because the death effect comes from the depressive effect, and the effectiveness of it comes from the analgesic effect. So not much more to it than that. You just get a chance to see the therapeutic index at work when we're talking about the different effects of the same drug. And again, that's an important thing to note, that each effect of a drug is going to have its own dose response curve. And if the effect of the drug ends in death, then its dose response curve is the upper end of the margin of safety. Relating to this is the therapeutic window. So the therapeutic window is basically where you have to keep the drug above minimum effective. So when you're talking about someone who's maybe bipolar, and you need to keep it above the minimum effective so that they don't have major mania or depressive symptoms. So you got to keep it above that minimum effective line here. But you don't want to go above the toxicity level. So what they do is they test this out, and they try to find the exact dose and frequency of dose that's needed to stay between those two levels. Let's look at potency next. So potency is going to look at basically the dose of a drug needed to achieve a given effect relative to another drug or a standard. And the important concept of this is affinity. So affinity is the readiness with which two molecules join together. Things that have a higher affinity are going to have more potency, because the molecules are going to join together easier. Relating to that though, drugs that are more potent need a lower dose. Drugs that are less potent need a higher dose. And when we look at the things that are on this slide, we have to consider how much of the drug, how much of a dose do you need to get the desired effect. When we look at something like acetaminophen, it has a very long curve, meaning it is less potent. You need more to get an effect. However, a thing to consider here is these ceilings, but the ceiling effects, the ceilings of a drug are not related to potency. It is possible that a drug cannot, a high enough dose, a high dose of a drug can never reach the effect of another drug, but that doesn't change its potency. That isn't related to potency. So the most desirable drugs are going to have a high affinity for sites of action producing therapeutic effects, and low affinity for sites producing toxic effects. So when we're talking about affinity, we want them to have we want the dose to be as small as possible to get that high action producing therapeutic effect sites. So sites that get the thing we want, high affinity there, and low affinity at the sites that produce the toxics, so we can keep the dose low enough that it doesn't affect it there. Let's look at those ceiling effects though. And that is the thing we were talking about earlier, which is effectiveness. And this is the maximum effect of a drug irrelevant of dose. So when we were looking at potency on the previous one, and acetaminophen was down here like this, that basically came down to, and that's not exactly how it is, but the point is its ceiling effect is much lower, so it has lower efficacy. Okay, let's transition now into some things relating to repeated administration. So we're going to talk about some of the effects of repeated use of drugs. The first and most important is what's referred to as tolerance. You should have already know this, but tolerance is where there's a decrease in the drug's effectiveness. This is where it takes more and more of a drug to get the effect. This is actually seen in many of the drugs that are commonly abused, that you need to keep more and more, you need to keep upping your dose to get the same effect. However, the reverse of that is sensitization. Sensitization is where drugs become more effective. And then there's the middle of this, which is neither, where there's neither tolerance or sensitization. So sensitization, where drugs become more effective. This is rare. Actually, typically the only time you get sensitization, and it does happen, is with the adverse side effects. But then the ones that have neither, this is also important. Why is this important? Because, as I said before, most drugs have multiple effects. Most drugs do multiple things. So when we look at opiates, they give the analgesic effect. If I can speak, they basically are a painkiller. There's more to it than that, but they're basically a painkiller. They're a number. And they've got that, and they've got the effect of suppressing respiration, depressing respiration. However, the painkiller effects have tolerance, meaning the effectiveness of the drug decreases over time. You need to take more and more of it. The depression of the respiratory system does not have tolerance. It might even have sensitization, or it's got neither, but the point is it doesn't have tolerance. So as you have to keep increasing the dose to get the painkiller effect, it is getting you closer and closer to the effect of killing you, because it depresses your respiration and heartbeat. So that is something to consider, is when you're looking at multiple effects of a drug, one effect might have a tolerance, another effect doesn't. And this is typically what leads to overdoses. There's also withdrawal symptoms that will occur if an individual stops taking a drug. This is using some of the same mechanisms as tolerance. So this is what indicates a physical dependence. So what's going on here is the body adjusts to you taking the drug. So the body tries to stay in stasis. And you taking a drug puts the body out of stasis. So the body stops doing things to naturally produce things relating to that drug. So all of the drugs are relating to neurotransmitters. So if you get a drug that increases the release of a neurotransmitter, the body will naturally start to decrease the production of the neurotransmitter. So even if the release is increased, there isn't as much to release, so it kind of averages out. And that's actually what causes the tolerance effect, because the body is counteracting a lot of the effects of the drug, so you have to take more of it to get over that counteraction. Well, when you stop taking the drug, those effects then cause withdrawal symptoms because now you have too little of that in the system. And this it can cause illness, all things like that. So it's one of those where it comes back where you get a physical dependence, and it's one of the things that keeps people using drugs. Let's talk about placebos and the placebo effect very briefly. Everybody in here should know what that is, so it's not something that's new to you. But placebo is where an inactive substance can have a physiological or psychological effect, typically relating to belief that it is going to cause an effect. But recent studies actually have shown that even if you know the placebo effect, and even if you know you're being given a placebo, it can actually have an effect. So it's not just in the range of belief that's causing it. It's subconscious things are going on. And it can result from changes in motivation, expectation, learning. It helps investigators investigate behavioral effects of drugs on humans. It can be used in studies with lab animals. Basically, it used to be used more than it's used now. It's actually used less now. And why is that the case? When we're talking about drugs now, most effects that we're looking at, let's say we're looking at painkiller or something like that, most effects have a gold standard. Most effects that we're looking at drugs for have a gold standard, meaning we've got studies showing that this one specific drug does this to this effect. So instead of comparing people to placebos now, they have the control group actually get the drug that's the gold standard to see if the new drug does better or worse than the gold standard. That is a better way of doing it because then you're getting rid of the placebo effect there. The fact that they use it in lab animals, they do that more to keep everything the same. So if you're giving a pill to one animal or an injection to one animal, you have to give a pill or an injection to another animal, or you've changed essentially the behaviors, what's being done to them. We're not going to talk about placebos more than that, just that it's an interesting effect in psychology. Okay, from here on out, we're going to look at the effects of these things on the production, the effects of these different drugs on the production of neurotransmitters. So most drugs are going to affect behavior. Most drugs that affect behavior are going to affect the synaptic transmission. So we talked about that before, the synaptic gap, the neurotransmitters that go across the synaptic gap, how they bind to receptor sites, how they're released from the presynaptic cell into the synaptic gap. They bind to sites on the postsynaptic membrane. All of that fun stuff we talked about before. Okay, these drugs we've been talking about today are going to affect that. They're going to affect it in one of two ways. They're going to affect it in an agonist way. Agonist drugs facilitate the postsynaptic effects, so they essentially increase effect. And then they also affect it in antagonist way. These inhibit the postsynaptic effect. These reduce it. So agonists are intending to increase the postsynaptic effect. Antagonists are intended to reduce the postsynaptic effect. There are many different ways. There is a long list of ways, 11 specifically. There's more than this, but there's 11 main ways that drugs affect the synaptic transmission. There are 5 that are antagonist, and there are 6 that are agonist. So the blue ones, 1, 4, 6, 9, 10, and 11 are agonist. They increase the postsynaptic. The red or orange boxes, depending on the color of your screen, the 5 of those are antagonist. They reduce. And if we went through these, I could go through these. 1 and 2 up here basically relate to the production of and transport of neurotransmitters. One, which is a agonist, it basically increases the production of neurotransmitters. Two, inhibits the production of the and transport of the neurotransmitters. Three is basically going to work on storage. It's going to basically prevent them from being stored in the vesicles. 4 is going to basically stimulate the release. 5 is going to inhibit the release of the neurotransmitters. 6 is going to stimulate the postsynaptic receptors, and 7 is going to block the postsynaptic receptors. So you see that there's agonists and antagonists that are doing opposite effects at most of these stages. One of the ones that's a little bit different is that 11 is an agonist effect that actually results in 11 and 10 are kind of the same thing. They both are going to result in the neurotransmitter staying in the synaptic gap longer, which is going to cause the postsynaptic membrane to fire more. 10 blocks reuptake, so the neurotransmitters stay out, and 11 blocks metabolism, blocks the neurotransmitters being broke down. And then it goes on from there. You've got the autoreceptors being blocked or stimulated. These are just all these different agonist and antagonistic effects. You're not going to have to know each of these. Just know that for most things, there's an agonist and an antagonist effect that work against each other. So if the drug is an agonist, it will cause an increase. If it's an antagonist, it'll cause a decrease. In a lot of them, it's the same on both sides. We're going to actually look at most of these, though. So an agonist is pharmacologically defined as a drug that causes a change in receptor activity. There's two different types. There's direct. This is going to mimic the effect of a neurotransmitter. So direct agonists, these are ones that are going to basically bind to sites and do what a neurotransmitter does. Then there are indirect, which are going to do a bunch of those other things we looked at. These are going to enhance the release of neurotransmitters, block reuptake, block metabolism, enhance binding affinity, basically making it so binding sites more easily are bound to, that type of thing. Let's actually look at some of these in slides, in various different GIFs. So first is the direct agonist. This is one, so like an endorphin, it fits in, it causes a reaction at the site. Morphine is a direct agonist in that it does the same thing. It will fit into the same receptor as the endorphin, and it will cause an increase in firing. So that is a direct agonist. Let's look at some of those indirect ones now. Indirect. How about one that enhances release? So you've got one here, normal, you get some release. Now you get an amphetamine, and you get a large release. How about an indirect agonist that blocks reuptake? So normally these would be sucked back up in. However, if you're using cocaine, it blocks that reuptake, so those stay in the gap, keep firing going on, so it can't be pulled back in, so it causes firing to occur again. How about one that blocks metabolism? So normally you've got metabolism that comes in, these enzymes, they come in, they get rid of the neurotransmitters in the synaptic gap. However, let's say you're looking at a drug that blocks that. So those enzymes don't go into the synaptic gap, and then it results in firing again because those neurotransmitters stayed in the gap. Let's look at one that's a little bit more complex. This is one that, again, increases binding affinity. So you've got GABA that opens up an ion channel. Valium comes in, and it basically results in the ion channel being open for longer. So it's enhancing binding affinity. Couple other things, two other things relating to agonists. First is partial agonists. These are drugs with a lower efficacy compared to a full agonist, but have the same mechanism of action. So we've got heroin here, but then you've also got buprenorphine, I'm not sure how to pronounce it, that does it at a lower effect. Sometimes you'll get this with drugs that are designed to treat withdrawal. So they still do some of the effects, so it still affects the withdrawal, but you're not getting the same side effects. And another thing that's usually done with these things that help with withdrawal and help with treatment of a drug addiction is these other ones might not build tolerance like the original does. The other one is an inverse agonist. So this is a drug with the opposite effect of a typical agonist. And in a sense here, what we're looking at is a drug that increases postsynaptic activation, but it decreases activity level. So when you increase drug concentration, even if it makes the cell fire more, that cell actually firing more causes a reduction in activity level. Final thing we're going to look at here is, before we look at some things with drug addiction, is antagonists. So this is pharmacologically defined as a drug that does not cause a change in receptor activity. So it causes the receptor activity to either remain at baseline or receptor activity to not increase. It basically, receptor activity does not decrease. It just doesn't increase. And there's two types here. First, there's competitive antagonists. These compete with a direct agonist or neurotransmitter for the same binding site, so they're basically going to these binding sites and blocking them. They're blocking the agonist or neurotransmitter from binding, and they don't cause the effect of the binding site. Then there is non-competitive antagonists. These are ones that do things like prevent release of neurotransmitters, alter the conformity of the binding site but don't bind to the binding site itself, or block the ion channel, so plug the ion channel. Let's look at those. First one, competitive antagonist. So heroin, it binds, it causes effect. Narcan is a drug that is given when somebody is overdosing on Narcan, or on heroin, sorry. Narcan will go in there, block it. The heroin then can't bind with it. The Narcan doesn't cause the effect, but the heroin can't bind with it. And in this case, it keeps the neuron from firing and can stop an overdose from occurring. So when we're looking at competitive antagonists, the dose response curve, we're looking at the fact that an agonist alone has a specific response curve. An agonist plus the antagonist moves it farther to the right, and again, that's where you're looking at the effect of that therapeutic effect. It moves the level of adverse side effects from happening farther to the right, so it decreases the chances of things like overdoses occurring. It might increase the concentration of agonists, but it keeps that concentration of the antagonist constant, and the agonist that eventually will end up outcompeting the antagonist for the same binding site. So that's one thing to consider. If the agonist remains the same, but the antagonist, if both the agonist and the antagonist remain the same, then eventually the agonist will outcompete it. So you have to either drop off the agonist or you have to increase the antagonist. So in our previous example with heroin and Narcan, you have to stop the administration of heroin, or you have to up the Narcan to end up counteracting it. Let's look at a non-competitive antagonist preventing release. So normally you've got release going on. Botulinum is there, and it prevents that from being released, so it doesn't fire. How about a non-competitive antagonist that modulates receptor conformity? So GABA opens up the ion channel. However, the picotoxin that binds, and it basically makes it so the receptor can't bind. So the receptor can no longer, GABA can no longer bind. It won't allow the ions through. The next, you've got, let's say, glutamate for a non-competitive antagonist that blocks the ion channel. Glutamate binds, opens up the ion channel, lets some through. However, let's look at ketamine. Ketamine blocks that, so even though the glutamate binds, the ions cannot get through because the ion channel is blocked. So when we're looking at a dose response curve here, we're talking about how the increase in dose of an agonist will not overcome a non-competitive antagonist, because the non-competitive antagonist is basically binding to a different site and blocking it. Increasing the concentration of an agonist but keeping the concentration of antagonist constant is going to result in the agonist not being able to overcome the antagonist, because, like I said, they're binding on different sites. It needs to wait for the antagonist to wear off before the agonist can have an effect. All right, let's finish up just with some on drug abuse and drug addiction. So drug abuse is a pattern of drug use that results in harmful consequences that is different than drug addiction, because drug addiction is related directly to a dependence. A tolerance has been built up and a dependence. So cessation of the drug will result in symptoms of dependence. It could be a physical dependence, it could be a psychological dependence, it could be both, and it results typically in loss of control of drug use that can and typically does result in harmful consequences. So addiction is the severe form. Drug use is using. Drug abuse is a pattern of using that results in harmful consequences. Drug addiction is dependence. So when we look at the substance use disorder in the DSM-5, you have to have 6 or more of the criteria. It's a mild substance use disorder with 3, 4 or 5 are moderate, and 6 is a severe substance use disorder. So we've got taking the substances in larger amounts for longer than you meant to. This is due typically to tolerance. Wanting to cut down or stop, but not managing to. Spending a lot of time getting, using, or recovering from the substance. Cravings and urgings to use the substance. Not managing to do what you should at work, home, or school because of substance use. Continuing to use even when it causes problems. Giving up important social, occupational, or recreational activities because of substance use. Using the substance again and again even when it puts you in danger. Continuing to use even when you know you have a physical or psychological problem that could have been caused or made worse by the substance. Needing more of the substance to get the effect you want, this tolerance, and development of withdrawal symptoms, which can be relieved by taking more of the substance. Okay. So, done talking about drug abuse and drug addiction. If you feel you might have a substance use disorder, there are a lot of hotlines out there you can get. The university has resources for you. I'm not going to go into it more than that. Just I'm not going to harp on you. Just going to say if you've got a substance use disorder, there are systems in place to help. Okay, in this lecture we covered an overview of psychopharmacology. We looked at pharmacokinetics. We looked at drug effectiveness. We looked at the effects of repeated administration. We looked at placebo effects very briefly. We spent a lot of time on agonist and antagonist with some interesting animations. And we finished with addiction. And then on Thursday, we will cover the actual neurotransmitters that are covered in this chapter. Thanks. Come on back.